Well, current presentation, we have the advantages and disadvantages of the online world. The advantages is that we have two speakers from the US today that can attend our lunchtime seminar in Europe, and the disadvantage is that we have some technical issues, but we are about to solve them. So I very much welcome you to the series, to the first seminar we have. We all know that basement membrane extracts play an important role in modern research. So we have great advances in stem cell technology, 3D models, organoids, and so on, and we consider them as strategies to replace animal experiments and to provide human relevant research. But at the same time, we have these animal-derived components in many of the protocols, and we know that this is not consistent with the aim of animal welfare, plus it also harms the aim that we want to have a pure human system. So there is a need to find replacement strategies for these BMEs, and we will have a set of presentations on these replacement strategies. Before we start with the presentations, I'm supposed to give some formal introductions. First of all, the session will be recorded and it will be made available on the NC3R website by the end of this week. And secondly, you will have a Q&A function in your Zoom, and please be encouraged to ask questions there. And I, as the chairman, will read these questions to our speakers. We will have a Q&A session after each of the two presentations. So our first presenter will be Simone Rizzi. He received his PhD at the prestigious ETH Zurich and then moved to Queensland University in Brisbane, Australia. In addition to his scientific life science degree, he also holds a master in industrial management, technology, and economics, also from the ETH Zurich. He was chief scientific officer at QGel for many years, where he was active in developing in vitro cultures based on synthetic hydrogels. And just recently he moved to a new company and is now working for Electromedical Systems, a medical device company. So my question to Emma and Simone is whether we solved the technical issues and can start the presentation.
I'm just waiting for the slides at the moment. Yeah. So we're having a few issues because they're quite large. Simone sending them to me. So yeah. Hopefully via WeTransfer in a second.
Yeah. I'm sorry. WeTransfer.
Strategies, all the opportunities and challenges. And Glen and Alekasnder will then also already give us some more detailed presentation on the different fields of BME application. I mentioned a few keywords already, and they will also talk about possible alternatives. So this will be very exciting as soon as the technical issues are solved. And the next sessions will then discuss some more specialized topics in the field. So please, yep.
Coming. I need to increase the code. Okay. To very important innovations, and also how now synthetic and defined alternatives are coming up in getting results that are very similar to what we get with BME. And essentially, when we talk about BME and replacement, in general, we think about always on culturing cells ex vivo, so outside the body, 3D cell culture, for instance. And this is in every form. So it can be bioprinting, it can be cells encapsulated within a gel, overlay culture, and all this type of different culture system. But essentially, if you boil down the culture of cells in 3D environment, as I mentioned before, we come to three main elements here. And the first element is the gel matrix that we are talking today about. This, they're naturally derived and artificial ones, but also you have a cell culture as well that plays a key role here. And you have the cells themselves. What is important here, and that would be my main point of this of my introduction talk, is that you need a context. You need a context when you are, thanks a lot. So you can go to the second slides. You need a context when you grow cells outside the body. We know that, for example, when we have cells inside the body, they are in a jelly matrix. They are exposed to theta factor from the ECM. They're exposed also to soluble factor, and also they need nutrients. And so this is where the media comes into play. And this is important that we make the context right in order to grow the cells ex vivo and they replicate some physiological features. So you can go to the next one, please. And when you get the context right, what happens is that you open up a number of applications where you can use those culture systems. And for example, you can have in basic research, which is mainly where BMEs are used to understand biological processes, how drugs work, to understand how diseases are forming and so on. But then you can apply also this culturing system towards more industrial application. And here I'm talking about drug screening application that have some industrial flavor. But as you move up this triangle, you can go into industrial application that have implication in the clinics. And this, they open up or they enter a new type of world where it's regulatory related. And here it's an important aspect to consider, and where perhaps, and I will talk more about that later, a replacement will play a key role. So let me go through in the next couple of slides to highlight a few examples where the use of BME has taught us a lot about what this context, the importance of the context of culturing cells that is required for culturing cells in 3D. And then I will transit to the artificial matrices, where I'll be talking about what are the latest developments that might play a key role in that. So BMEs, I checked a few papers. I think the first paper were described the production of BMEs was in 1977. And between then and 1977 and 1992, there were a lot of few labs that are using these matrices for some very interesting aspects. And here I choose this paper from the group of Minabi cells, where essentially that meant quite a lot for me, and also in the field that where I working previously for that type of application. So basically, by putting cells in 3D in a context, you can differentiate rapidly or distinguish between healthy cells and cancer cells, for example, malignant cells, just by the way they grow or the morphology they use. If we stay on the same group and a little bit on the same topic, another aspect that was very interesting here is the morphology, the phenotype of the cells. So you have normal cells. If you go to the next slides, please. Thanks a lot. So here the context is playing also a key role. You see healthy cells, they have a certain morphology, they form acini, they grow arrested. If you look on the middle images, is malignant cells, you see that they grow and have morphology that are irregular. However, when you perturb the interaction between the cells and the ECM, you see that you can revert those malignant cells to a more healthy phenotype. And this is also an important discovery. Please, you can go to the next slides, where essentially, despite the fact that malignant cells and the reverted phenotype have the same genetic background, they have a different phenotype, and also they behave differently. The growth is different, and so on. So this is, I like the importance of the context that I was mentioning before. If you come now to one of the latest developments in relation to the use of matrigel or BMEs, is organoids. And perhaps most of you are familiar with this. And also this is, I think, a key event where methodology were identified to grow single stem cells into basically a more organized 3D structure, and those stem cells can differentiate then into the different specialized cells that form the origin of the stem cells. And this is have implication, important implication, not only in understanding the developmental biology, but also in regenerative medicine, and also in diseases, as this methodology for healthy stem cell also, and please go to the next one, went into diseases such as cancer, such as cystic fibrosis. And I think we have a speaker that will speak about all these aspects using this methodology. And a decade after this discovery, this paper came out that essentially described that it is possible to grow cells, cancer cells outside the body, test drug on those cells that essentially were the same drug that the patient received, with astonishing results where basically in 100% of the cases where the drug did not work ex vivo on the patient cells, it didn't work in the patient. So this is very interesting potential application that go behind the basic research. And here now with time, this the use of BMEs and the methodology basically spread over different organs, different diseases, and this is what it was, in my point of view, a big learning from that. So if you go to the next slides, please. Yeah, so those are the different applications that I mentioned to you, just a few examples in different organs. Now, the next slides, basically the question I have here, so what is BME? So BME is essentially this is basement membrane that are ECM protein that are purified from a murine tumor. And this murine tumor is essentially inoculated in a mice over the limb. And this, when the tumor is growing, is purified, and you get this BME. If you go to the next slides, what we have seen in this example, that basically BME is we can say that is a one-size-fits-all. We see in the stem cells, disease cells can grow in this, and so on. So this is a matrix that really can be used for different applications. The drawback, and probably if you go to the next slides, please, is it composed, come back more. Is it composed by different proteins? So in a publication, they look at different batches, production batches, to see that there are over 1,800 unique proteins that identify in different batches. And if you go and you look at the normal, so what you purify or the growth factor reduced, there are also some proteins that are essentially the same, but they could find proteins that were not present in one or the other. This is not only coming, obviously, from different batches, different composition, but also, for example, when you measure protein content in the growth factor reduced, you might identify protein in very tiny quantities that you're not able to identify in the normal one. But the essence of this is that the composition of these BMEs is not defined, and we know and is also well documented in the literature, that you find some batch differences. So what are now artificial matrices? So now I would like to switch a little bit and transit to the artificial matrix. Please, the next slides. Here, the situation is completely different. The artificial matrices, they're composed not even 10. I think I never seen or never produced matrices with 10 components, but below 10 components. So there, you know exactly what components you are in there. They're well characterized and so on. So here in the artificial matrix, you use kind of a minimalistic approach where you have to identify which minimal components are required to be able to grow or induce the growth or the physiological feature of a specific cell type. And here you see the difference that you have design flexibility, you can change, which you don't have with BMEs, because what you get from the mouse is what you have. In contrast, synthetic and artificial, you can change. And you have defined composition, and therefore also very precise batch and no batch differences. You can go to the next one, please. So how are they composed? And I think here the next two speakers will go more into details, but in a nutshell, we, for example, copy what is in nature. So in nature, you can extrapolate different aspects of the matrix. So you have biological characteristic, biophysical characteristic, so the stiffness, the biological integrin binding sites, for instance, and biochemical characteristics, so the ability of the matrix to be remodeled by cell activity. So then what you do, you look at what are those domains, and you can extract them or synthesize them using chemical synthesis or DNA, recombinant DNA technology and other methodology. And then you can start from a polymer, inert polymer, and construct the biological, biochemical, and biophysical characteristic. And then using mild cross-linking reaction, you see at the end, you can bring all together in a hydrogel system. So in the next couple of slides, I would like to show you and give you a good news. Yes, it is possible to produce components or artificial matrices that can replace BME. Please, can you go to the next one? Thank you. So the first publication I choose an example is from 2016, but don't misunderstand me. The research in developing artificial matrices started far before that. This is probably now we kind of get the fruits from all this research and in terms of replicating physiological characteristics with artificial matrices. And this is essentially a publication that was recently published, and it shows that it is possible, as the case we saw before, from a single stem cells to produce an organoid that also have all the specialized cells in a 3D culture without the use of other cell types and so on. And this is just by identifying the right biological, biophysical, and biochemical components of the matrix. So this is one example. Then I would like to show you another example in the next slides, and where I was involved here. And it goes a little bit beyond this, because here we took patient tissues, and this was in collaboration with Herlev Hospital, and we have also a speaker in the next section from Herlev, Dr. Sidling. And here we took cells directly from tissues, and we identify artificial matrix composition that were able to expand those cells ex vivo in a similar manner that was previously shown with Matrigel. And also the differentiation of these cells basically show the fact that the cells remain stem cells, and they could differentiate into the different specialized cells of the colon. The next slides. And again, another good news in terms of replacement of Matrigel. Here is also a work that was involved in collaboration with Charles River, where we get the PDX-derived cells, so the patient-derived xenograft. So it's essentially cancer tissues, human cancer tissue that is grown in mice, and a model that are used as preclinically in drug development, for oncology application. So we received the cells that were grown in mice, so the human cells, and we cultured them ex vivo, and then we tested drugs ex vivo on samples that were grown in artificial synthetic gel and in BME, and then we compared the drug response between in vivo and in vitro. And what was very interesting here is that with the synthetic gel, we could basically predict in 91% of the cases what was going on in terms of drug response in vivo. And the control experiment we did with BME, they were a bit lower, about around 82%. So this is was to just summarize that BME has told us quite a great big deal, the use of BME in terms of 3D cell culture and things, but it is possible to find alternatives and to develop alternatives for this. And the research of alternatives, as I mentioned, started far before the example that I showed you with in the late 90s, in the 90s, 80s. Here I can mention the work of Professor Jeffrey Habel, where I work with, or Professor Glen Prestwich that he will be presenting later on. And basically now what we see today is the fruits of this research. So to conclude, if you could go to the next slides, where it shows some examples that are coming up on this replacement quite rapidly in the last few years. And here I would like to conclude my talk, perhaps the next slides, please, to come back to the triangle that I've showed you before, and where I put BMEs and artificial matrices head to head somehow. And here, I think in terms, when we speak about basic research or preclinical drug development, I think there is an opportunity of BME. For artificial matrices is a challenge, as we have seen. You need to identify the right compositions for that. So, and there are an increasing number now of expert, biomaterial expert in the research in academia, but also as a company that can support that. But, you know, there is not a one size fits all. But if you go to the next slides, if you combine this aspect with the design flexibility of artificial matrices, I think we get a lot far. And it's a huge opportunity also in basic research, where basically with artificial matrices, you can fine-tune, test one aspect after the other, and you can really also understand mechanism of the cells of diseases that are not possible to be seen with a matrix that does not have this design flexibility. But please, the next slide. If you move up the ladder, though, you will see, if you go towards industrial application or clinical applications where regulatory will play a key role, I think there the artificial gel, the artificial matrices have a huge, huge opportunity there. And of course, because of the inherent characteristic of BMEs and other natural matrices there, I see rather some limitations. So this is a little bit what I wanted to present to you, to somehow initiate a little bit this webinar series. And my next slide here is just the outline of the program of the next sessions, where the next one, the second one, will be about BME replacement in the research, basic research domain. And then the third one will go and tackle a little bit BME replacement within clinical application. And then the fourth section is basically a panel discussion around all these topics. So I would like to thank you for your attention. And also I would like to hand over the floor to the other speakers with their very interesting talks.
Okay, thank you, Simone. This was a fantastic introduction to the topic we want to discuss in these seminars, lunchtime seminars. Thank you so much for this. Maybe I can immediately ask a question. So, of course, this was extremely convincing, but I want to put a critical question. So, you stated that half of the truth of a cell lies outside the cell. And then you also presented that a natural matrix may contain 1,800 or at least a large number of components. And the synthetic matrix usually contains less than 10 components. So can this really reflect the natural physiological situation? 10 components, can they really reflect the situation, the physiology we have in a natural organ?
Yeah, this is a very good question, and it actually goes down to the essence of this. But I can make another question. Is those 1,000 proteins are those really required, for example? And if you grow cancer cells, you can grow cancer cells, you can grow stem cells. So are those cells requiring the same environment? I don't think so. So you see, there is also, if you look at the other side as well, you might not need all these elements. And we have seen as well that those elements might also inhibit some physiological aspects, you know. So now to come from the synthetic part and just a couple of words, what I'm saying is that with the synthetic, few synthetic messages that you give the cells, you kind of give the first input to the cells. And then while they're growing, the cells, they are actually expressing also their own matrix, their own environment. And this is basically where they grow, and then they can express then their physiological features. So this is also something that we need to take into account, that you might give the first stimulus for the cells to grow into a certain aspect. Of course, you have positive aspect and negative aspect, as you said, is still an artificial matrix. It's still an in vitro model, you know, when we look at both sides. And there are pros and cons for that. They remain a model at the end of the day.
Sure, sure. So we have a question from the audience, from Bernard. He asked, did you ever try to combine BMEs and artificial matrices? For example, supplement a low concentration of BME with a single artificial component? Would that make sense? Did you ever try this?
I think we tried for saying that, okay, our type of proteins that are in there, for example, laminins are important, or different collagens. Yes, it is possible, and it was tried. The question is that you might not solve the problem. You know, what problem are you trying to solve? Do you want to find a replacement? Or do you simply want to look at a certain biological phenomenon in basic research where perhaps the BME are enough and you don't need a synthetic matrix? It is important, though, to reflect at the beginning of your research where I want to go. Can I, with the model that I'm using, can I go to the end of where I want to go? Or I have to switch model at certain point because, I don't know, regulatory, because I want to do a clinical application, then I cannot use BME. So then if that is the case, I would really reflect and start already from the beginning with the right model, you know, either replacement or a BME aspect, a natural matrix. So these are questions, fundamental questions. The tendency of scientists perhaps is to look also where I get the most comparison, you know, and of course there is a lot research done, paper published with BMEs, and you won't find perhaps for your application for synthetic, which then makes the challenge to compare. What's my benchmark, you know? So those are a little bit aspects that need to be thought when you start doing your research.
So we have another question that comes from Kathy Mary. she writes, thank you, Simone, great introduction. Yes, I agree. In your experience, what are the major challenges for groups when it comes to uptake adoption of synthetic matrices? Sorry.
A very good question. It is true that when you tackle the synthetic, the artificial matrices, you might have to pair up with specialists on a specific gel type or matrix type, because as we have seen, there are many aspects that has to come together. You know, the synthesis, the characteristic of the gel, how to test it, what type of chemical reaction you use, and so on. So I think this is an important aspect to consider. Unfortunately, today, there are not many out-of-the-shelf solutions that you can take and use. This is a little bit unfortunate, but I think this will change with time, will change this aspect. So the main challenges are really who can provide me with the right elements and teach me how to use these elements in my research. And even further, how can I change this element to create new matrices, because the first matrix that you will try, you might not be the right one, you know, so you want to change, to trial and error, you know, and this is an important aspect. I know it's not easy, and the chemical expert, the biomaterial expert are also looking for biology competences, and perhaps the biologists, if they want to move into this, they might have to look for some chemical bioengineering competence in this area.
Okay, so thank you. For the sake of time, we should soon move to the second talk, and maybe I ask the last question for your presentation that was posed by Maren Hülsemann. She writes, might be answered in the session in 2 weeks, but from the practical side, what does a researcher need to do who wants to switch to the artificial base? How much work is it to find the minimal requirements components? Can I compare my old experience still with the ones in the new system? That adds up to the last question. So maybe you can give a brief answer on some suggestions what researchers have to do.
Okay, again, you need to identify which matrix you use, and then related to the matrix, artificial matrix, you might have some partner that can come on board with you. Could be an industry partner, or could be even a research groups. But there are a lot of groups now that are researching on this. Now, there are ways. We had a microenvironment screening. We have a microenvironment screening methodology where we could screen hundreds of different grow conditions in one single experiment, and within 2 or 3 weeks understand what gel and media composition was best for this cell line. This is kind of the brutal approach where you screen with some, as you say, some guess that come from, you know, what cell type, what type of elements you need to have in that. Or you can do by trial and error in a small scale, you know. This is the two different approaches. And in the literature, you will find methodologies that describe that in this. Yeah, I know it can be complex, but again, I think with collaborations with groups that have developed this type of things, I think it's could be very promising. And they will probably also very happy to see where are the problems that they can solve, and they can generate or produce an artificial matrix to solve this problem.
Okay, so thank you, Simone, for your presentation and your answers. I think we now move on to the second presentation. So the second presentation will be given by 2 speakers, and I'm very thankful to both of you because you woke up so early. The presentation will be given by Glen Prestwich and Alekasnder Skardal. Glen is distinguished professor at Washington State University. He has a long history in this type of research. He was co-founder of 12 startups. He's fellow of the AAS and the National Academy of Inventors. And I think it's an impressive number that you have more than 600 publications and almost 50 patents. This is really a lot. And so the second speaker, Alekasnder, is from the University of Utah. He works, he's now at the Ohio State University, but he received his PhD at the University of Utah, where he worked on ECM-derived hydrogels for 3D bioprinting. And he is now also founder and director of the Organoid Technology Program of the Ohio State University. And the title of your shared presentation is Presenting Different Fields of BME Application and BME Alternatives. So thank you for joining our lunch seminar, and the screen is yours.
Thank you very much. Alex and I have organized it that I'll give the introductory materials, and then Alex will go more deeply into bioprinting on organoid and more recent applications. So if the slides are able to come up on the screen, then I'll start off our session, our part of the session. This has been extremely great to have Simone, Dr. Rizzi, give his introduction to the environment, and I think the way we fit in really dovetails nicely to the extremely elegant and thorough introduction that he gave. So let's go to the next slide, please. I'm going to talk about the how and the why of BME replacements, and there are really three takeaways. First one, please, should just show the translating research that we're talking about is really our jobs as researchers. We're not just there to play around in our lab. We're there to do something to make a difference. The next little bullet is that if we're going to translate stuff in medicine, it's complex, it's expensive, and slow. You've seen some of those, some of that in Dr. Rizzi's slides. Next slide, please. And the fact is, as he said also, successful products are simple, and they're the ones that give clinically meaningful outcomes. So in answer to the very last question, the important questions to ask are the ones that are going to save people's lives. Next slide. So Simone introduced this concept that extracellular matrix is necessary for regenerative medicine. A cell needs to have some information, some contact, something to sit on and do its business. And you can either do that with a complex cell instructive matrix, such as shown on the left, or you can do it with a simple matrix, such as the Alekasnder products from many years ago, with just hyaluronic acid and gelatin cross-linked together, as shown on the right. Next slide. So how do we know what we want to do? And this was really what this first session is about. What are the biological criteria for these materials that are going to be effective BME replacements? Well, we really want to create a temporary being the key word, synthetic extracellular matrix environment with the right mechanics and chemistry. We want to use that to deliver cells and molecules to the right place. We want to keep them there. We want to keep them healthy, let them grow, differentiate, and create a new tissue, and let them create their own tissue-appropriate ECM. Then that will facilitate tissue regeneration and, in principle, achieve a clinically meaningful outcome. That's the biology of the problem. But the next slide, please. Yes. But the actual problem is more complex than that. There's a huge number of hurdles that we have to get across, from clinical need and intellectual property to preclinical, clinical, business, financial, product design, and ultimately reimbursement. And I will come back to this theme later. But let's go how this translates into a new set of design criteria, since many of us are from an engineering background. What are the design criteria that we need? In the real world, we have to meet the needs of the end users, who ultimately will be the patients and physicians. And we have to give them something to use that's simple, safe, and effective, and easy to use by the physicians at the end, in the final process of administering it to a patient. It's got to be affordable and scalable to make under current good manufacturing practices, manufacturing processes. It's got to have a robust intellectual property portfolio, or else nobody will invest in it. And there needs to be a viable business model. The cost of goods, the difference between the cost of goods and producing this stuff and what you can sell it for has to be enough to make it attractive for industry. There needs to be a clear regulatory path, and there must be reimbursement available by healthcare payers. I can tell you that there are a lot of approved drugs and devices that are not reimbursed, and if you don't pay for it out of your own pocket, it doesn't get used. And so the healthcare payers are really the final arbiters of what gets into the actual clinical setting for patients and physicians to use. Next slide, please. So we started working on this many years ago. I always like to say I was dragged kicking and screaming into the world of glycosaminoglycans in the late 80s when a student came back to my lab to work on it, and I had no idea what it was. But HA is really a wonderful material for everything that we know about in biology, everything from hydration and permeability and lubrication and embryogenesis. But now it's become incredibly attractive as a source of commercial products. Next slide, sorry, the next image shows why that's the case. It's because hyaluronic acid actually has just a few sites of modification that can be easily manipulated. And rather than go into every single one of those, I'm just going to talk more generally about the BME part of this. So I'll skip past the chemistry part to the next slide, please. And I'd like to categorize the kinds of modifications that you can make on HA into monolithic, where you make a physical change, a chemical change in the molecule, and then all you can do is change its physical form, to what is new, which is the living form of HA, where you can do what Dr. Rizzi was talking about, which is customize the material so that you can customize the shape, the size, the composition, when it cross-links, what the mechanics are, what the degradation rate is, whether or not you add cells or drugs before or after gelation. A lot of things can change. And so the old products, not the old, they're current products, Synvisc, Monovisc, Gel 1, all injectables for osteoarthritis, Restylane, Juvederm, all injectables for dermal fillers, as well as Elevest. So it's a really interesting group of materials now that we can get into, that are now into the clinic in some cases for cell delivery and cell therapy. And that's where we're going with this. Next slide, please. So the materials that were developed first in my lab, patented, and then by Glycosand, which then became Biotime, involves the cross-linking of HA that's been modified with the gelatin, which has also been similarly modified using thiol Michael addition chemistry. And that has allowed us to go into all the same areas as the monolithic materials, but with a lot more control. And in the next slide. This was also the basis that we used for making materials that were the very early versions of 3D culture and 2.5D culture, if you will, on a sponge, and the very beginning of what was going to be like a BME replacement. And Alex will talk a little bit more about that. But this was this is one of the very first slides I used to describe to researchers why plastic was not the best place to grow cells, and how you could grow cells readily on an HA gelatin matrix without all that much additional trouble. Next slide. Ultimately, we need to get these BME replacements out of the laboratory and into the hands of physicians and into the bodies of patients to fix things that are broken. And so the business strategy has actually been a barrier in accomplishing this, because not only do you have the intellectual property of the BME replacement, but now you have the intellectual property of the stem cells or the cells that have been derived out of a particular lineage and have IP around them. Now you have to combine these two, and the industry, the two companies, have to figure out how to play nicely together and both make money. And so that often is unless there's an acquisition so that both things are in the same company, that can be a barrier to translation. Next slide. Nonetheless, this roller coaster ride to the clinic is ongoing, and I put this slide up for a very good reason, which is that this was I was asked by Science Translational Medicine editors to identify a handful of key leaders in the field, including the directors of laboratories at MIT and director of the California Institute for Regenerative Medicine at the time. This was back in 2012. And you can see that the same problems that we have now are the problems from well, 9 years ago. And it's difficult to figure out how to get materials to the clinic. It's still expensive, it's still time consuming, and there's still a lot of unknowns that have to be navigated. So next slide, please. Nonetheless, this regulatory roller coaster is leading to a few products in the clinic now. This has been described now both in the literature and in seminars that the Renivia, which is a CGMP version of HiStem, has now been used in the clinic to deliver liposuction-derived stromal cells for HIV-induced lipoatrophy with incredible 100% basically success in the patients that receive this for rebuilding their facial features. And so that's a win. It's now, there's a CE mark for this for cell delivery in the face in Europe, and it can be used off-label for other uses. Next slide. That same material is now being developed in a slightly different format for patch grafting by Lola Reed's group and a very large team both in the UK, in Europe, and the US. And that first paper has come out now and several more are coming, and there's some intellectual property barriers and investor barriers that are being navigated. So translation is not for the faint of heart. It's not just working in the lab and discovering something exciting and new. It's getting past a lot of hard-headed people who don't want you to succeed sometimes. And that's a challenge that you have to face if you're going to be in the entrepreneurial or the industrial world. And then the last example I want to show is one that really is at the true cutting edge of what's going on. This is from Khalid Shah's group at Harvard, Brigham and Women's, and Khalid has started a company called Amasa, and Amasa has now acquired all of the remaining Renivia from Biotime and is getting ready to go into the clinic to use essentially HiStem to deliver engineered mesenchymal stem cells into the cavity created by resection, removal of a glioblastoma in the brain. And so they're going to be healing brains and reducing metastatic brain cancer by using cell therapy using a BME replacement, a very simple one, just 2 components plus a crosslinker. So that's the last part of that slide. Next slide, please. So my final slide is about what I call the translational imperative, which I hinted at the beginning, which we've got to embrace this incredible complexity of what we're trying to accomplish, going from 2D to 3D and multiple tissues and multiple types of things that can be added and so forth to engineering a versatile solution and having a versatile solution that can be something that meets all needs. But still can be customized. And then, but ultimately, we have to deliver something simple. If you can't make a simple product that people can just say, oh, I'm going to buy that one, then it's really hard to get this stuff into the clinic and making a difference in people's lives. So with that, I'll say my thank yous, and then I'll transition over to Alex, who will go onward with his presentation. And this is the obligatory disclosure slide, because I have been involved with a number of companies that I've started or work with to commercialize things based on chemically modified glycosaminoglycans. Thank you very much. And Alex, the floor is yours.
Alex, I think we can't hear you. Probably your microphone is muted.
Yeah, I just found it. Can you hear me now?
Maybe you can repeat what you started saying. Thank you.
I didn't say anything yet, but anyways, thank you. All right, so we'll dive right in. First, got a couple of financial interests that I'm supposed to disclose in matrix diagnostics and Excel biologics. But to dive into the real stuff, when we talked last week in preparation, Jens asked if anybody knew how much Matrigel is used throughout the world. And I don't know that that figure is exactly known, but there are some market statistics that show that, the global 3D cell culture market, is estimated at 1.5 billion. It seems maybe a little low, I don't know, but the growth is expected to be huge and to be somewhere, between 4 and 11 billion, which is a huge range within a few years. As of a few years ago, Matrigel was believed to have 70% of the market share, and basically accounted for over 95% of most publications that use 3D cell culture. And then the revenue is huge. Now, there are some other BMEs, but especially coming online in the last few years, but Matrigel is the big elephant in the room. But because even though this is like the product that everyone uses, and my PowerPoint is on autopilot, so excuse me if I have to backtrack a little bit. The composition precludes it from a lot of clinical translations. And this includes tissue engineered products that would go into patients, 3D cell culture platforms for cell therapy or like expansion of cells, and also clinical diagnostic assays, organoids for precision medicine diagnostics or prognostics in the clinic. And it's due to the composition. These are murine derived, they're from tumor tissue, and they're a black box of various factors. So we've always been wondering, can we design something better? And so Glen mentioned HiStem. It's had a number of names over the years, but basically this hyaluronic acid and gelatin that's cross-linked. And after I left Glen's lab, one of the first applications we tried to apply some of these materials to was culture of hepatocytes for drug screening. And so 10 years ago, that was a couple years before everyone figured out it was actually pretty easy to grow primary human hepatocytes as spheroids with the right cell culture media. But our approach was, can we actually use sort of the materials that are native in the liver, essentially decellularized extracellular matrix as a supplement within these simplified ECMs. And what we found was when you compare these liver ECM hyaluronic acid hydrogels to the standards that were being used, collagen 1 sandwiches, for example, these ECM containing HA gels are able to maintain sort of the hepatocyte cell-cell interactions much better with increased viability and function. They can allow for real-time drug metabolism that can be recorded, and that's only in the groups that have these extra ECM proteins added. And this is, again, due to probably this another black box, admittedly, of potent growth factors. So we wanted to see if we could strip this down even more and get further away from the black box of growth factors. And so but we also thought that sort of the HA and gelatin system for some applications might be too simplistic. So what we've been doing over the last few years is moving to using collagen, since cells remodel it into different fibrillar architectures that can be super important in various disease states. We manipulate different cross-linking approaches to enable things like bioprinting, which I'll touch on the next slide. We have a modular chemistry strategy where we can link in synthetically modified adhesion proteins like fibronectin, laminin, etc., through thiol maleimide interactions or thiol acrylate interactions. We can also put in growth factors if we want, and then use the cross-linking to control the mechanical properties. And so this essentially resulted in a material that, through a variety of cross-linking approaches, creates a thixotropic, essentially a material that responds to physical stresses on demand, where in this graph here, what you're seeing is an increase in the shear stress that you would see during, say, bioprinting. And when you reach a particular magnitude, the material fails because reversible bonds break. And down here where the G double prime is greater than the G prime, it behaves as a liquid. But if you remove the stresses, aka after the material comes out of the bioprinter, it sets back up, it bounces back, and you have a solid. And so this actually makes it a really nicely printable material, where now you can essentially plug and play these materials into any bioprinter with very little end user customization. So these are a number of commercially available bioprinter platforms where we've not had to actually change the formulation. It's I won't call it off the shelf, but it's probably about as close to an off the shelf bio ink that there is, that has the complexity of a stripped down extracellular matrix. So we've deployed this material in a number of applications, which I want to highlight a couple to show that you can do quite a lot without needing BMEs. So one of these examples is taking organoids and combining them with microfluidics to study aspects of disease such as cancer metastasis. So we've talked about, Simone mentioned organoids and cancer organoids a little bit, but with relatively simple technologies, we can model, say, placing a tumor in one of these locations, introducing flow, and different organoids, also supported by the same type of material. And now we can actually watch labeled tumor cells grow in a primary tumor site, jump out into the environment, and because this is under flow, eventually these cells can travel through the circulation and graft in these different organoid sites. And so this is we've published on this a couple times, and we're still building on it. We're also working sort of in the neural space, which is not something I expected to be doing about 5 years ago, but we've become interested in the blood-brain barrier through our work in tumor organoids, specifically gliomas. But the blood-brain barrier is a big part of many diseases, MS, dementias, etc. There's a lot of trafficking that happens between other parts of the body and the brain, microRNAs, exosomes, etc., that might play a role in these diseases. So we expanded on sort of the pretty common transwell model, where normally you've got monolayers of cells on the transwell membrane, and these are okay, but we wanted to add sort of a three-dimensional flavor. So we've used our matrix in the transwell to create this three-dimensional space where you can sort of model the brain parenchyma. So in our simplest system, it's just astrocytes and parasites with endothelial cells on top. But with this hydrogel, which we can again tweak with laminin or fibronectin to kind of mimic the brain ECM, we get really nice endothelial barrier function, which can hold tier levels with tight junction proteins are expressed, efflux pumps are present, all important to the blood-brain barrier. But where we're going today is, again, adding microfluidics to it and making a more modern version where we actually have essentially a blood vessel on a chip that's built into the brain parenchyma. And you can see here this sort of structure. So we're really excited about this. We're in the works of trying to place patient-derived brain tumor organoids in this platform, which brings me to sort of the last several slides, which, again, we've talked about organoids, and Simone highlighted some papers from the Cleavers group and others. And I think in this space, I like to highlight this New York Times article from a couple years ago, which questions how successful, precision medicine really is, or precision oncology. Specifically, the process where a patient would have a biopsy or a piece of their tumor that's resected, sent off usually to some third-party company for genetic sequencing, which can identify mutations that might be actionable for targeted drug therapies or chemotherapies or immunotherapies. In reality, what often happens is, and I've seen these reports that come back to the clinic, usually you have multiple drug types that might be usable. And so it's still upon the oncologist to determine sort of a best guess treatment. And if you look at the actual stats, I need to find an updated paper probably from the last year or two to add to this. We really don't see a huge increase in successful treatments in patients. Sure, it works for some patients, but precision medicine has not really become the silver bullet that it was touted to be. And so obviously, we're not the only people doing this, but the idea is, can we plug our organoids into this into this workflow to find a better therapy? And most of the folks who are doing this are doing it with basement membrane extracts, Matrigel. The work from the Cleavers group and others is fantastic. The amount of data they've accumulated, the gastric cancer paper that Simone highlighted is a fantastic paper. But then if you go to the methods section and you look at not just the fact that they're using Matrigel, but look at the cell culture media formulations, it's got like 100 different components in it. It's not DMEM, it's hard to standardize. So we've taken our relatively simple ECMs, hydrogels, and we've successfully deployed them to create patient-derived organoids in mesothelioma, lung cancer, glioma, colorectal, melanoma, appendiceal cancer. Now we're working in several adrenal or endocrine tumors, sarcoma, blood cancers. So it is possible to use these simple systems. And I want to highlight glioblastoma for a second. And there's a lot of, these heat maps have a lot of things going on, but basically what we did was make organoids from glioma specimens from 6 different patients. That's the ACEFGH. And we're comparing the original tumors, organoids, or treated organoids versus plastic after culture. And the way that a unbiased hierarchical cluster analysis of the top 1,000 variably expressed genes comes out is that they tend to cluster with tumor, organoid, treated organoid for several patients. And then you have, for a number of patients, just plastics cluster together. So this shows that even our simple ECM system is able to maintain the genomic profile of the original tumor, whereas as soon as you go to plastic, you have genomic drift. Now, the question probably someone's asking is, have you compared it to Matrigel? We have not. We want to. And where I want to end this is just with a small anecdote, which is when we after we'd been presenting this over the last few years, everyone came back and asked, well, what about immunotherapies? Can you do immunotherapies? You need an immune system. So we've been taking lymph node tissue from the same patients that we get the tumors from, and the lymph nodes have cells representative of about 80% of the immune system. And so when we introduce those cells into these organoids, only when we have those immune cells present and treat with, say, immune checkpoint inhibitors, do you get cell killing or T cell mediated tumor cell killing. So in here we've got red is dead, green is viable. And to end this, we had, well, not me, but my collaborators had a patient presenting with melanoma on one of his arms, a kind of armpit, shoulder area. And he had gone through first treatment with pembrolizumab, second treatment with ipilimumab. These are incredibly expensive immunotherapies. At least in the United States, these can run hundreds of thousands of dollars to $1 million for like a full course. I assume in Europe, maybe the insurance is better. But basically, what we did is we created tumor organoids, these immune enhanced organoids, and also found that these checkpoint inhibitors did very little. But one of my students had been reading up on melanoma and said, hey, let's try this other therapy, and dabrafenib and trametinib, and it actually killed more than 50% of the cells within a couple of days. So what we ended up doing was, well, my collaborator, ran down the hall with the data to the oncologist and said, you got to get this patient on trametinib or dabrafenib. But his initial sequencing did not have the right mutation identified, so they couldn't do it. But our persistence allowed the cancer center to send out a new biospecimen from his tumor to the sequencing company, which came back with a MEK mutation, which qualified him for trametinib, and he started using it, and within 3 weeks or so, the tumor shrank, and he called the oncologist and said, hey, my arm is better, I can move it. So that sounds like kind of like a miracle, but trametinib, when it works, is incredibly potent and fast. But he became resistant to the therapy and the tumor rebounded. We actually made organoids again from his new tumor or recurrent tumor, and it was now also resistant to this therapy. So he died, but I think this is just a little anecdote or case study that shows that these organoids, whether they're BME based or not, have a lot of potential. Now, I think basically I've shown that you can do a lot with these well-defined ECM hydrogels, these different models that I've shown. But I will admit that our system does not have the same potency as Matrigel. We don't do long-term propagation of patient-derived organoids. So there's still a huge opportunity and commercial opportunities to develop better alternatives that have the same potency as Matrigel. And the question that Glen and I have been asking is, can we use a bioengineered human cell-derived platform as a better product? And we're doing this today. And it's using the cells to do the work, not the chemists. And it's called Humagel. And I wish I could talk more about it today, but it'll be a few months until the patent gets filed. So I will end it there and thank all the people that actually did all the work in my lab and obviously the funding. So I will jump off the screen share, and I think if there's any questions, happy to answer them.
So thank you, Alekasnder, for this fascinating presentation. And of course, Glen also, thank you for the first half. I have a question. When you talk about your HA gelatine hydrogel, what is the origin of the gelatine?
Very good question. Originally, it was bovine, and as as the mad cow disease thing came in just about the same time that this is developing, the company Biotime switched 100% to porcine. So it's porcine gelatin, and it's made under CGMP conditions, so that Renivia is a sterile CGMP product.
So, but it's still of animal origin. So is it still possible to use this in humans?
Yeah, porcine gelatin is commonly used in a lot of different products for tympanostomies and everything from tympanostomies to surgical meshes.
But would it be an advantage to have recombinant human collagen converted into gelatin?
Yes, it would, but there we run up against cost of goods problem. The amount of money that you would have to put in to do that and then develop it as a product and pay for the production of recombinant human gelatin, human collagen, and then have to denature it, which seems kind of kind of a bad thing to do to perfectly good human collagen. That actually never met the financial criteria for becoming a product or becoming something you could develop as a product.
So we have a question by Kathy Mary. She writes, thank you. It seems to me that the real challenge here is create systems where cells grow in 3D in a relevant setting, but in a micro format which is fast to get results back to the clinic. Do you see the same thing? What do you think are the challenges for this?
Yeah, no, that's a good point and sort of question. That's actually how we tackled this, was less as a research tool, and at least for our patient-derived organoids, less as a research tool that we were, again, going to propagate for multiple passages for months and months, and more how fast can we determine what therapy might be best for a patient. So what we've published for a number of cancer types is essentially we have drug screening data back within a week or two. So that would be actionable data that could be used in the clinic to help make decisions. And if you compare that to current precision oncology practices, when you send samples out to, Tempus or whatever company's doing your sequencing, Foundation Medicine, another one, you're talking usually a month or so before you get the data back. So organoids can beat sequencing analysis in terms of speed of, getting data back that you can make decisions. Now, ultimately, we're not trying to replace sequencing. I don't want to give that opinion, but I think if you add these things together, you have more data that can help the oncologist. Also the point about a micro format, I didn't focus in on this, but one of the videos that I had the bioprinting going was printing organoids into a 96-well plate. And so we can create hundreds of organoids from patient samples within a few minutes using bioprinting. And we're trying to expand to sort of 384-well format, et cetera. And now you're in sort of the high throughput realm where, paired with the right plate reader or high content imager, you could, basically transition everything to high throughput, which is great.
Good. So thank you again to both speakers for your presentation and for answering the question. There is one more question, sorry. Thank you very much. Great presentations. How difficult was it to establish the lymph node cultures? Did you try this in several patients with reproducible results?
Yeah. So we didn't really do lymph node cultures per se. It was more that we dissociated node tissue and used those cells and integrated them right into the tumor organoids. And that, again, we called it sort of immune enhancing the organoids. it's a little artificial in a way, right? Because normally you'd have node and tumor, not node and tumor together. But this comes down to the speed aspect. So how fast can you get the results back to make a clinical decision? And if you have them separate, you need to wait for cells to traffic back and forth. If you put them together, it's faster. So again, clinical, little artificial, which we acknowledge. But yes, we did it for multiple patients, both in melanoma, as I showed. We've published it with, I think, 9, 10 patients. We're working on a manuscript right now with a whole bunch of sarcoma patients. We've done it in appendiceal cancer, and it's been not that difficult. Now, I will say again, with our ECM, that simple but supportive helps. And then then we have the heterogeneous environment. So we're not just sorting for tumor cells. We're taking all of the cells that are in the tumor. They're probably all helping support each other. So again, letting the biology kind of do the work is probably part of why it works as well.
Yeah, I think the heterogeneity is really important, that having the heterogeneous tissues and letting the biology dictate. I mean, the cancers are heterogeneous. Even a given tissue has a lot of different cell types in it, and you need all of them to really to have the biology be properly recapitulated. So.
Okay, so we have 2 more questions. Maren Hülsmann asked, or first wrote, thank you very much, fascinating work. Did I understand Alekasnder correctly that organoids are not doing too well on the artificial BME for a longer time? Why is that?
So hopefully I didn't misspeak. So no, I think if you look at the literature, organoids generally do better on BMEs like Matrigel for long-term cultures. I am 100% transparent that we have not tried to go long-term, again, because we came at this from a clinical point of view where we wanted to turn data around as fast as possible to make decisions. So we're just now starting to see how far we can culture them in our stripped down matrices. I don't think we'd be able to compete with Matrigel in terms of, at least right now, with our current system, to go months and months and months, right? So there are folks in the community who say it's not an organoid unless it's been passed, unless it can be passaged 5 times. I completely disagree with that, like arbitrary classification, but yeah. So no, our material right now is not potent to the degree that BMEs are. But that's what we're working on, is can we find some sort of middle ground?
Thank you for this open answer. And we have the last question from Christopher. How much available do you think that human tissue will become available for in vitro studies? Are human biobanks being developed in the US or in Europe? What constraints you believe there are? So this is about the problem to get enough human tissue for these studies.
I mean, I can start, and if Simone or Glen have their thoughts, feel free to jump in. At least in the US, Glen kind of alluded to this, that it is cost prohibitive to like have human tissue probably just to derive ECM. There are some companies doing it, but it's for research materials, not materials for clinical use. In terms of like in vitro models, I don't know that there's huge biobanks being made, but there are biobanks being made at academic institutions. But it's right now, at least when I was at Wake Forest previously and at OSU now, it's largely investigator driven. So they're kind of smaller biobanks or programmatic biobanks for research uses. But human tissue for building in vitro models, at least in my experience, has not been a problem to get, to obtain. But then I have colleagues across campus who can't get tissue at all. So it's all about networking and getting in touch with the right surgeons and that sort of thing, in my opinion. So I don't know that there's a big system yet, but there's plenty of tissue.
Okay, so pretty much ran out of time, so I think we should come to an end. Thank you to the speakers again and to the audience for staying in the seminar for so long. Emma, maybe can you quickly put up the schedule for the next presentations? We will have the next seminar in 2 weeks. It's again on Thursday. It's on November 4th, same time. And we'll have 3 speakers then. Maybe if you scroll down a little bit. Yeah, now you can see the next session, session 2, BMEs and replacement from a research perspective. So we will have Kathy Mary as the chair. We'll have 3 speakers, Andreas Kurtz, my colleague from Berlin, Irini Veliu and Jennifer Ashworth. And I hope to see so many attendants again next time. And I'm not sure if the organizers have anything else to add. Otherwise, I would thank everybody for being in this lunch seminar and wish you a good afternoon.
Yeah, we'd just like to say thank you to everybody for joining. We will be getting the recording online if you'd like to share that with colleagues later this week. And yeah, hope to see you on the 4th of November. Thank you all.
Thank you.
Okay.
Thank you.
Thank you. Bye-bye.
Bye-bye.
Thanks.
Thank you.